everyone. So question number 1 is from the chapter Organisms and Population, class 12, and the question is, in spite of interspecific competition in nature, which mechanism the competing species might have evolved for their survival? So we know that in any natural environment, no single species can live in isolation. In fact, they survive through interspecific interaction. So suppose this is species 1, this is species 2. So some kind of interaction will exist between them. Now this interaction can be beneficial or positive interaction. It can be detrimental or negative interaction, or it can be a neutral interaction. Okay, but here we have to name the mechanism that the competing species have developed to survive without disturbing each other. Okay, and such a mechanism is called resource partitioning. So what happens in resource partitioning? See, suppose there is a tree in an area and some bird species live on this tree, but after some time another species of bird arrive here and they also want to live on this tree. But these two species don't want to fight with each other. So what will they do? They will do resource partitioning. That means they will distribute the areas of this tree into different zones, and then they will reside in their allocated zones without disturbing each other. So this way they will coexist independently. Okay, so the correct option will be resource partitioning. But let's discuss other options as well. So option number B is competitive release. What does competitive release mean? See, suppose there is a species of organism which live on a very small area because its competitively superior species occupy a larger area. Okay, now these less fortunate species are unable to compete with their superior competitors, so they are confined to this small area. Okay, now they can be released from this area only if somehow we remove the superior species. Okay, so this is what happens in case of competitive release. These superior competitors are experimentally removed and therefore the competitively inferior ones get released from the small areas they were confined into. Okay, now let's come to the third option. So the third option is mutualism. Okay, so what is mutualism? It is also a type of interaction where two organisms coexist. Okay, but here they don't live independently. In fact, they help each other. So species 1 will help species 2 and species 2 will help species 1 in return. So this interaction will be beneficial for both the species. Okay, now the last option is predation. What is predation? It is a kind of interaction where species 1 eats the other species. Okay, so here it is a beneficial interaction for one while it is detrimental for the other. Okay, and the other species eventually gets eliminated. Okay, so this was the explanation of this question. Question number 2 is from the chapter Anatomy of Flowering Plants, class 11. And here we have to match list 1 with the correct option in the list 2. Okay, so let's see number A. It is written cells with active cell division capacity. And the correct match for this is meristematic tissues or meristems. Okay, what are meristems? See, the word meristem is basically derived from a Greek word meristos, which means divided. So these cells are able to divide rapidly and therefore they are found at the tips of shoots and roots because these regions need to grow continuously. Okay, so here you can see the first option is the only one where it is shown that 2 is the correct match for A. So you can easily mark the correct answer here. But we'll discuss other statements as well. So let's see number B. It is written tissues having all cells similar in structure and functions. And the correct option is simple tissues. Okay. So what are simple tissues? See, as the name suggests, simple tissues are made up of only one kind of cells. Okay, therefore all these are similar in structure and functions. Okay, now these can further be divided into three types: parenchyma, collenchyma, and sclerenchyma. Okay, so what is the basic difference between them? See, parenchyma are very thin walled tissues and they are composed of cellulose. Okay, collenchyma on the other hand have thickened corners. Okay, and why are these corners thick? It is due to the presence of cellulose, hemicellulose, and pectin. Okay, so collenchyma is composed of cellulose, hemicellulose, and pectin. Now the last one, sclerenchyma, are generally long and narrow cells with thick walls and they are composed of lignin. Okay, so this is the common difference between parenchyma, collenchyma, and sclerenchyma. Now sclerenchyma can further be classified as fibers and sclerids. Okay, out of these, sclerids are dead cells and they have very narrow lumen. Okay, so here you will see in the question, statement D is talking about scleres. Here you can see option D. Here it is written dead cells with highly thickened walls and narrow lumen. So it is talking about scleredes. Okay, so this is the correct option for this. Now what is left? Option C. So tissues having different types of cells. These are basically complex tissues and they are also known as vascular tissues. Okay, and vascular tissues are composed of two types of cells, two types of vascular bundles, xylem and phloem. Okay, xylem is used to transfer water and minerals, whereas phloem is used to transfer food. Okay, so this was the explanation for this question. So question number 3 is from the chapter Biotechnology, Principles and Processes, class 12. And the question is, during the purification process for recombinant DNA technology, addition of chilled ethanol precipitates out. So we know that in recombinant DNA technology, we cut out a DNA segment using restriction enzymes and then we ligate this fragment into a vector in order to transfer it to a host organism. Okay, but to cut this DNA, it has to be in its pure form such that no other macromolecules remain there. Okay, but what happens when a cell is cut open? All the macromolecules, including RNA, protein, polysaccharides, come out along with the DNA. So we need to remove these macromolecules. And how do we do this? See, RNA is removed after treatment with nucleases. Similarly, proteins like histones are removed after treatment with proteases and so on. And at last, chilled ethanol is added, after which DNA is precipitated out. Okay, so the correct option is DNA. So this question is from the chapter Transport in Plants, and here we have to match list 1 with the correct options in list 2. Okay, so the first one is cohesion. So what is cohesion? See, co means common or mutual. Okay, and the suffix hesian means to stick. That means when same type of molecules stick to each other, it is known as cohesion. And when we say cohesion, we generally talk about the mutual attraction among water molecules. So the correct option for A is 2. Okay. Now the next is adhesion. So what is adhesion? It is just the opposite of cohesion. So what happens in adhesion? In this case, different types of molecules stick to each other, okay? And it is generally referred to the attraction between water molecules and some polar surfaces, okay? So the correct option for this will be 4, attraction towards polar surfaces, okay? Now the next one is surface tension. So what is surface tension? See, this term explains that the water molecules are attracted to each other more in the liquid phase than in the vapor phase. Okay. Therefore, when you store water in a container in the form of liquid, it will stay there for a long period of time. But when it starts boiling and getting converted into vapor, it will start moving everywhere. Right? Why? Because mutual attraction is lowered. Understood? Now, the last one is guttation. So, what is guttation? See, what happens in plants, the roots exert a positive pressure due to which water moves upward. Okay, and one of the effects of this positive root pressure is the loss of water from the margins of the leaves in the form of liquid droplets. Okay, and don't confuse it with transpiration. What happens in transpiration? In that case, water is lost in the form of vapor, but here it is lost in the form of liquid. Okay, so what will be the correct answer? Option A. Got it? Number 5 is from the chapter Sexual Reproduction in Flowering Plants, class 12. And the question is, the term used for transfer of pollen grains from anthers of one plant to stigma of a different plant, which during pollination brings genetically different types of pollen grains to stigma is. Okay, so let's first understand how pollination works. As most of us know that a typical angiosperm flower consists of a male reproductive part called andrecium and a female reproductive part called gynecium. An andrecium consists of a large number of stamens, whereas gynecium is composed of one or more pistil. So here you can see I have already drawn diagrams of a typical stamen and pistil. Okay, so as you can see here, a stamen consists of a long slender part called filament and a terminal, generally bilobed structure called anther. Now, this anther is the part where pollens or male gametophyte are produced. Okay, and as you can see, the pistil consists of a stigma, a style, and an ovary. Now, what happens in pollination? Pollens from the anther of a flower are transferred to the stigma of a pistil. Okay, now depending on the way this pollen is transferred to the stigma, there are different terms given to the pollination. Okay, so let's see the first one. First one is where the transfer of pollens from the anther of the flower takes place to the stigma of the same flower. Okay, so this is termed as autogamy. Okay, that means pollens of one flower are transferred to the stigma of the same flower. Now, this can also be classified on two types depending on whether the flower is exposed or closed. Okay, if it is exposed, the process is known as cosmogamy. Okay, cosmogamy. Whereas if it is closed, the process is known as cleistogamy. Okay, so both of them are types of autogamy, right? Now, second type of process is known as getonogamy. What happens in getonogamy? The pollens from one flowers are transferred to the stigma of another flower. Okay, so this is known as getonogamy. But since this is taking place in the same plant, so there will not be any difference in the genetic of the progeny produced. Okay, now the third type is xenogamy, where the pollens from one plant are transferred to the stigma of another plant. So now this brings genetically different types of plants because there is a transfer between two different plants. Okay, so according to our question, the correct answer would be xenogamy because two different plants are used in xenogamy. Hello everyone. Question number 6 is from the chapter Cell Cycle and Cell Division, class 11. And the question is, which of the following stages of meiosis involves division of centromere? So what is meiosis? It is basically a special type of cell division in which the chromosome numbers are reduced by half, and therefore this is also known as reduction division. Okay, the meiosis is divided into two parts, meiosis 1 and meiosis 2. At the end of meiosis 2, we get 4 haploid cells. Okay, now meiosis 1 is further divided into 4 stages: prophase 1, metaphase 1, anaphase 1, and telophase 1. Similarly, meiosis 2 consists of prophase 2, metaphase 2, anaphase 2, and telophase 2. Okay, now what happens in prophase 1? See, the homologous pair of chromosomes form bivalents and then they undergo crossing over. Okay, now in the next phase, that is metaphase 1, these bivalents arrange themselves on the equatorial plate, and as you can see, two parallel plates are formed. Okay, here we must keep in mind that the bivalents consist of two sister chromatids, okay, sister 1 and sister 2, which are attached to each other at the centromeres. Now, this centromere is also important as the spindle fibers are attached to these centromeres, and with the help of centromeres, these spindle fibers pull these chromosomes towards the opposite poles. Okay, now each pole receives half the number of chromosomes of the parent cells. Okay, and then in telophase 1, what happens? Nuclear membrane and nucleolus reappear and two cells are formed. So we can say that metaphase 1 cannot be the answer because centromere is not divided at this stage. Okay, now let's consider the other stages of meiosis 2. See, in meiosis 2, similar stages take place. Okay, meiosis 2. Okay, what happens? Here you can see at metaphase 2, what is happening? The difference between metaphase 1 and metaphase 2 is that here single plate is formed, okay, at the equatorial plate. Here two plates were formed. Okay, now in the anaphase 2, the sister chromatids get segregated and the centromere is divided. So the correct option will be anaphase 2. Okay, what happens during telophase 2? See, it is the last stage when the chromatids reach the opposite poles and start uncoiling. Okay, so this was the explanation for this question. So this question is from the chapter Biotechnology Principles and Processes, class 12, and the question is, which of the following is a correct sequence of steps in a PCR or polymerase chain reaction? Okay, so we know that DNA is the genetic material which is responsible for transferring of genetic information from one generation to the other. Okay, and it consists of two strands of opposite polarities, right? Now, suppose we want certain characteristics to be incorporated in the next generation, then we can take the desired piece of DNA and then multiply it. Okay, so suppose this is the desired DNA. Okay, then we'll take it and synthesize its multiple copies in vitro, okay, using a set of primers and DNA polymerase enzyme. So how do we do this? We use the method of polymerase chain reaction. What happens in polymerase chain reaction? It consists of 3 steps. In the first step, a very high temperature is applied. What happens due to this? The two strands of DNA get separated, okay, and the DNA gets denatured. Therefore, this process is known as denaturation. Okay. Now, in the next steps, 2 sets of primers are provided. What do these primers do? These get annealed to the 3' end of the DNA strands. Okay. So these two are the primers. Okay. Primers get annealed. Therefore, this process is known as annealing. Okay. Now, in the third step, an enzyme known as DNA polymerase is provided plus some nucleotides known as deoxynucleotides are provided. Okay, so what happens? This DNA polymerase enzyme extends these primers to form two separate copies of DNA with the help of the nucleotides which were provided. So since the extension of primers takes place during this step, therefore this step is known as extension. Got it? So the correct option will be denaturation, annealing and extension. I hope you understand. So this question is from the chapter Plant Kingdom, class 11, and the question is Gemi are present in, and the options are mosses, pteridophytes, some gymnosperms and some liverworts. Okay, so what are Gemi? See, Gemi are small green and multicellular asexual buds. Okay, asexual buds, that means these can be present only in those plants which reproduce asexually. Okay, now so this gymnosperms and pteridophytes cannot be the correct answer because both cannot reproduce by asexual methods. Okay, so the only possible options are mosses and liverworts. Okay, so let's see what is common between mosses and liverworts is that both belong to the group of plant known as bryophytes. Okay. So what are bryophytes? These live on soil but they need water for their reproduction. That means they need water as well as soil for their survival. So they are also known as amphibians of the plant kingdom. Okay. Amphibians of plant kingdom. Now, bryophytes are classified into two groups, okay, namely liverworts and mosses. So, what happens in liverworts? Here, the body of the plant is in the form of thallus. That means it is not differentiated into leaves and stems, okay. It looks like leaves, but it is not the real leaf. Okay. Now on these thallus, we can find small receptacles, and inside these receptacles, you will find gemi. Okay. These receptacles are known as gemma cup. Okay. And inside the gemma cup, you can find gemi. Okay. So the correct answer is liverworts. What happens in mosses? Mosses has two stages of development. Okay, the first one is protonema and the second one is leafy stage. And how does it reproduce? The reproduction in this case is by the fragmentation and budding of the protonema. Okay, so this question is from the chapter Principles of Inheritance and Variation, class 12th, and the question is the production of gametes by the parents, formation of zygotes, the F1 and F2 plants can be understood from a diagram called, and the options are bullet square, punch square, Punnett square and net square. Okay, so what is a Punnett square? It is basically a diagram in the shape of a square. That means it is a kind of table. Okay, which can give us all the possible outcomes of a particular cross. Okay, that means if we have two parents, say P1 and P2, and we know the genotypes of both the parents. Suppose the genotype of the first parent is capital T, and the genotype of the second parent is small t. Okay, then with the help of the Punnett square, we can find the expected percentages of different genotypes in the offspring of these two parents. Okay, so let's see how do we do this. Suppose, see, this is capital T. So we write the genotypes of parent 1 here. Okay, capital T. Similarly, we write the genotype of parent 2 this side, small t. Now let's cross it. So capital T and small t will give us a capital T small t. Similarly, capital T and small t will give us a capital T small t. Again, small t, capital T. And again the same. Okay, so that means these four are the possible genotypes that will be present in the offspring that will come after the crossing between these two parents. Okay, understood? So the correct option is Punnett square. Okay. Question number 10 is from the chapter Evolution, class 12, and the question is the factor that leads to founder effect in a population is, and the options are natural selection, genetic recombination, mutation, and genetic drift. And the correct option is genetic drift. The explanation lies behind an important principle called Hardy-Weinberg principle. This principle talks about genetic equilibrium. So what is genetic equilibrium? It states that the percentage of occurrence of alleles of genes in a population remains constant through many generations. Okay. That means if a population has, say, gene 1, gene 2, gene 3, then in the next generation, the population will also have the same genetic setup. Understood? But this is the normal case. In some exceptional cases, what can happen due to some change, this genetic equilibrium might get disturbed, which can lead to a change in the genetic setup of the resulting population, which will eventually lead to the process of evolution. Understood? Now there are 5 factors that can affect this genetic equilibrium. One is gene migration and the other are 4 options that are mentioned in the question. Okay. So let's see what is a gene migration. As the name suggests, it happens when the population moves from one place to another. So what will happen during this process? Some of the genes might get passed to the new population, whereas some might get lost. Okay. And if this gene migration occurs accidentally or by chance, then it is known as genetic drift. Okay. So genetic drift takes place accidentally. Now this genetic drift can be of 2 types. One is bottleneck effect and the other is founder effect, about which the question has been asked. What is bottleneck effect? Suppose this is a population and it has 50 types of genes. Now when it goes to another place, the new population might get 45 genes which were present in the original population and 5 genes might get lost during the process. So what is the similarity between population 1 and population 2? This is quite similar to that. It is just slightly different, isn't it? This is slightly different population. So this is what happens in bottleneck effect. Whereas in founder effect, the new population is entirely different, or you can say entirely new species is formed because the new population has hardly 1 or 2 type of genes that were present in the original ones, while all the other genes are newer ones. Okay. So now you understood how genetic drift affect the founder effect. Why it is known as founder effect? Because the original population is the founder of the new population. Therefore, it is known as founder effect. Understood? So what is mutation? Mutation is something that happens randomly. Okay. As we saw here that genetic migration or genetic drift had some reason or certain method, but this mutation is very random. There is no particular reason for that or no particular process for that. Okay, it is a very random process. Now what is natural selection? Natural selection is basically survival of the fittest. That means the genes which are capable of surviving in adverse conditions will be able to pass to the next generation, whereas those which are weaker will eventually get eliminated. Okay. Now what is left? Next is genetic recombination. Okay. So this happens in case of gamete formation. Okay. So it happens during the gamete formation when there is some alteration in the normal process. So question number 11 is from the chapter Plant Kingdom, class 11, and the question is genera like Selaginella and Salvinia produce 2 kinds of spores. Such plants are known as, and the options are Homosaurus, Heterosaurus, Homosporus and Heterosporus. So here you'll see 2 types of words, sporus and saurus. Okay. Now the word sporus is related to spores, whereas sorus are the clusters or structures which produce spores. Okay, these are generally found at the lower surface of the leaf of ferns, and these are brownish or yellowish in color. Okay, so the options which contain this sorus are not correct. Okay, now out of homosporous and heterosporous, let's see which one is correct. See, the prefix homo means same, and the prefix hetero means different. So the plants which contain same type of spores are known as homosporous. One of the example is pteridophytes. Okay, most of the pteridophytes are homosporous. And the plants which contain or which produce more than one type of spores, they are known as heterosporous. So Selaginella and Sylvania are examples of heterosporous plants. Question number 12 is from the chapter Plant Growth and Development, class 11, and the question is, plants follow different pathways in response to environment or phases of life to form different kinds of structures. This ability is called, and the options are elasticity, flexibility, plasticity and maturity. And the correct option is plasticity. Okay. So let's see what is plasticity. See what happens during the process of development. Plants adjust their pathways according to different phases or environmental conditions. For example, if you look at these leaves, so these leaves are of the same plants called larkspur. Okay, but this is the juvenile leaf, that means the leaf that is found in the juvenile phase of the plant, whereas this kind of leaf is found in the adult phase. Okay, so can you see that this plant has changed the pathway and different type of these structures are present in different phases of their life. Okay, similarly, another example of plant known as buttercup is that on the land its leaves are more broader. Okay, whereas in the water medium its leaves are narrower and thin. Okay, so depending on the environmental condition, it has changed the shape of its leaves. Okay, so this ability of the plants to follow different pathways in different situation is called plasticity. So plasticity is the correct option. Okay. Question number 13 is from the chapter Biomolecules, class 11, and the question is, which of the following are not secondary metabolites in plants? And the options are morphine, codeine, amino acids, glucose, vinblastine, curcumin, rubber, gums. So let's see what are metabolites. So metabolites are those substances which are used in the normal metabolic processes, or you can say metabolism. So these are either produced or used up during the metabolic activity. Okay. Now these are classified as primary metabolites and secondary metabolites. So what is the difference between primary and secondary? See, primary metabolites are absolutely essential. That means these have identifiable functions and important roles to play in the normal physiological functions of plants or animals. Okay. For example, if we talk about amino acids, then we know that amino acids are the monomers which polymerize to give us protein. And protein is absolutely important for the normal growth and development of any organism, isn't it? Similarly, if we talk about glucose, then glucose acts as primary energy source, and no organism can produce its daily functions or daily activities without obtaining energy from the glucose. So out of the given options, we can say that amino acids and glucose are primary metabolites and not the secondary metabolites, whereas all the other options are the secondary metabolites of plants. That means they are involved in the metabolic activities of plants, but these are not absolutely essential, or these are not important in their physiological functions. Okay, so plants can survive without these secondary metabolites. Okay, I hope you understand. Question number 14 is from the chapter Molecular Basis of Inheritance, class 12, and the question is, Complete the flow chart on central dogma. So here you can see a flow chart representing a central dogma, and this central dogma was proposed by a scientist whose name was Francis Crick. Okay, and according to the central dogma, the genetic information flows from a DNA to an mRNA to a protein. Okay, now there are three main parts of the central dogma. Let's see what are they. So the first one is replication. So what do you mean by replication? See, the word replication means reproducing or duplication. That means during this phase, a DNA is duplicated, or you can say a DNA is produced from a DNA. So how does this happen? We know that a DNA has two strands. So during this phase, both the strands are separated. Okay. And new complementary strands are synthesized for each of the two previous strands, giving rise to two new DNA molecules. So you saw how new DNAs are produced from an initial DNA. Now, in the next part, that is known as transcription, the genetic information is copied from a DNA to an mRNA. So how does this happen? See, one of the strands of the DNA is used as a template strand by an enzyme known as RNA polymerase enzyme to produce an mRNA that is complementary to this template strand. Okay. Now the last part is translation. So what happens in translation? So during this phase, amino acids are polymerized to give protein. And how does this happen? See, if this is an mRNA, then a ribosome molecule binds at a specific location in the mRNA, and then they start adding amino acids one by one to give a polypeptide chain or protein. Okay. So out of the given options, option C is correct, as here you can see the copying or duplicating of DNA is represented by A, that is replication. Okay. Then the copying of genetic information from DNA to mRNA is transcription. So B is transcription. Now mRNA to protein is translation. So C is translation. Now at last what is formed? Protein is formed. So D is protein. Okay. So C is the correct option. Now other options have mentioned a word called transduction, which is not at all related to the central dogma. What is a transduction? It means the flow of genetic information from one bacterium to another bacterium with the help of a virus or a bacteriophage. Okay. So this is not a part of central dogma. Okay, I hope you understand. Question number 15 is from the chapter Cell, the Unit of Life, class 11. And the question is, when the centromere is situated in the middle of two equal arms of chromosomes, the chromosome is referred to as, and the options are metacentric, telocentric, submetacentric, and acrocentric. So let's first see what is a chromosome. So chromosome is a structure which is formed after the DNA is packaged with the help of a protein known as histones. Okay. Now, every chromosome has a primary constriction which is known as centromere. On both sides of centromere, there are disc-like structures which are known as kinetochores. Okay. Now, on the basis of location of this centromere in a chromosome is classified into four types. One is metacentric, second is submetacentric, third is acrocentric, and fourth is telocentric. So let's see what is the difference among all of them. In case of metacentric, the centromere is located right in the middle of the chromosome, forming two equal arms. Okay. Now in the submetacentric, this centromere is located slightly away from the middle. Okay. So it will form one smaller size of arm whereas the other arm being longer than the other. Okay. Now in the acrocentric, this centromere is present near the end of the chromosome, forming an extremely short arm and a very long arm. Okay. In case of telocentric, this is located at the terminal point of the chromosome. Okay, that means right in the end of the chromosome. Okay. So the correct option will be metacentric for the statement mentioned in the question, that is in the middle of two equal arms. Okay, got it? Question number 16 is from the chapter Biotechnology, Principles and Processes, class 12. And the question is, DNA strands on a gel stained with ethidium bromide when viewed under UV radiation appear as, and the options are yellow bands, bright orange bands, dark red bands and bright blue bands. And the correct option is option B, bright orange bands. So let's see. We know that one of the important steps in recombinant DNA technology is cutting of DNA with the help of a restriction enzyme or a restriction endonuclease. Okay, and then ligating it to a vector in order to transfer it to a host organism. Right. Now, once we cut the DNA with the help of restriction endonuclease, it results in the fragments of DNA. So some fragments may be small, some fragments may be big. So these fragments have to be separated using a technique known as gel electrophoresis technique. So what is gel electrophoresis? Here we take the DNA fragments, and since we know that the DNA fragments are negatively charged molecules, so these can be separated by forcing them to move towards anode, that is positive cathode, under an electric field using a matrix or a medium. Okay. Now one of the common matrix used today is agarose. Therefore the process is also known as agarose gel electrophoresis. Okay. Now once the DNA is separated on the basis of size, that means smaller the DNA, the farther it will move. Okay. So once we get the separated DNA, these can be visualized only by staining them with special compound known as ethidium bromide compound and then exposing them to UV radiation. Why? Because we cannot see pure DNA fragments in the visible light. Okay. So these will appear as bright orange colored bands. So what was the role of ethidium bromide? It basically acts as an intercalating agent. Okay, intercalating agent. That means they are stacked in between the nitrogenous bases of the DNA and then make them visible. Okay, so I hope you understood. Question number 17 is from the chapter Plant Growth and Development, class 11. And the question is, the site of perception of light in plants during photoperiodism is, and the options are shoot apex, stem, axillary bud and leaf. And the correct option is option D, that is leaf. So let's see what is photoperiodism. It is basically response of plants to the duration of light or darkness. Or you can say response to a period of day or night. So how does this happen? Let's see. There is a well-defined critical duration in some plants. What is critical duration? It means that there are certain plants which require exposure to light for more than this critical duration or below the critical duration. Okay. So this is suppose above the critical duration and this is below the critical duration. Now, depending on how much exposure a plant needs, they are classified into long day plant and short day plant. Okay. So what is the difference between long day plant and short day plant? Long day plants need exposure above the critical duration. That means if they are exposed to light for more than the critical duration, then flowering will take place. But if they are exposed for less than the critical duration, then no flowering will take place. Okay. Similarly, in short day plant, just opposite will be the case. That means If they receive light for more than the critical duration, then flowering will not take place because they require exposure for lesser amount. Okay, that means they need exposure for lower than the critical duration. So in that case, flowering will take place. Okay, and the point of perception or the site of perception of this light is the leaf. Okay, so the correct option will be option D. Now there is a similar process which is known as vernalization. What happens in this phenomena? Here the flowering does not depend on light, but it depends upon the exposure to low temperature. Okay, so in this case the site of stimulus is shoot apex. So if it would be asked about vernalization, then the answer would be shoot apex. But in photoperiodism, the answer is leaf. Okay, I hope you understood. Question number 18 is from the chapter Biotechnology and its Applications, class 12. And the question is, when gene targeting involving gene amplification is attempted in an individual's tissue to treat disease, it is known as, and the options are biopiracy, gene therapy, molecular diagnosis and safety testing, and the correct option is option B, that is gene therapy. So what is gene therapy? It is basically a collection of methods that are used for the treatment of hereditary gene defects. That means those gene defects that are present from the time of birth. Okay, so let's see how is it done. Suppose this is a cell or a tissue which consists of a defective gene which is responsible for carrying the disease. So in this process, a healthy or a normal gene is inserted inside this cell to take up the function of this defective gene. Okay, so this healthy gene will get inside the cell, multiply itself and perform all the functions that this defective gene was unable to perform, so that this cell can act or function normally. Okay, so this was about the gene therapy. Now what is biopiracy? Biopiracy means unauthorized use of bioresources. Okay, that means suppose there is a bioresource, it can be anything, be it rice or any other. Okay, and it belongs to a certain country, and any other country or multinational companies wants to use this resource, then it has to take the permission of the country of origin or follow certain procedures such as payment of compensation in order to use these bioresources commercially. But if some countries or some companies do not follow this rule and do not pay the compensation, then they are said to be involved in biopiracy. Got it? Now what is molecular diagnosis? It is a method of early detection of pathogens or disease in order to understand the nature and the causes of disease. Okay. Some of the examples are PCR, ELISA, recombinant DNA technology, etc. Okay. Now what is safety testing? See, safety testing is also known as toxicity testing. It is basically a process in which transgenic animals are made more sensitive to toxic substances so that the effects of these toxic substances can easily be studied. Okay. Question number 19 is from the chapter Plant Kingdom, class 11, and the question is, which of the following plants is monoecious? And the options are Carica papaya, Cara, Mercantia polymorpha and Cycas circinalis. And the correct option is option B, that is Cara. So let's see what is meant by monoecious. See, the word monoecious has been made up from two different words, mono, that means one, and a Greek word called oikos, that means house. So if more than one things are located at the same position, it is said to be monoecious. Okay. Similarly, there is another word dioecious, which is made up of two different words, di, that means two, and oikos, that means house. So what is dioecious? If two or more things are located at different positions, two different positions, then they are said to be dioecious. Okay. Now in biology, these words are referred to the position of male and female sex organs. So if male and female sex organs are located on the same plant body, then the plant is said to be monoecious. Okay, so Kara is the example of monoecious plant, since most of the species of Kara are monoecious in nature. Okay, on the other hand, if male and female sex organs are located on different bodies, then the plants are dioecious in nature. So these options, Carica papaya, Mercantia polymorpha and Cycas circinalis, are dioecious in nature. Okay. Question number 20 is from the chapter Biotechnology and its Applications, class 12. And the question is, which of the following is not an application of PCR or polymerase chain reaction? And the options are molecular diagnosis, gene amplification, purification of isolated protein, and detection of gene mutation. And the correct option is option C, that is purification of isolated protein. So let's see what is a PCR. It is basically a reaction that is used to produce multiple copies of gene or DNA. Okay, and it consists of three steps. Number one, denaturation. In this step, a very high temperature is applied so that the two strands of the DNA get separated and the DNA gets denatured. Okay. In the second step, that is known as annealing, a primer is annealed or attached at the 3' end of the template strand. Okay. And the third step is extension, in which the primer is extended or amplified. Okay. Now, if these steps are repeated many times, then multiple copies of genes can be produced. Okay. Now, what are the applications of PCR? First of all, we amplify a gene, okay, so that it can easily be detected. So, this option, gene amplification, is one of the applications of PCR. Now, why do we amplify gene? So that it can easily be detected. So it helps in early diagnosis of any kind of disease. Okay. So molecular diagnosis is also an application of PCR. Now, if we are able to visualize the gene clearly, we can also see any kind of change or mutation taking place. So detection of gene mutation is also an application of PCR. But purification of isolated protein is not at all related to PCR, since PCR is involved with nucleic acids or DNAs and not with proteins. So this is an odd option. So this will be the correct option. Question number 21 is from the chapter Cell, the Unit of Life, class 11. And here we have to match list 1 with the correct option in list 2. So we know that a eukaryotic cell consists of a well-defined nucleus containing chromosomes, right? And the cell also consists of various organelles which are useful to the cell in many ways. So one of the important organelles of the cell is mitochondria, which is also known as powerhouse of the cell. This mitochondria is a double membrane structure consisting of an outer membrane and an inner membrane which forms infoldings. So these infoldings are known as cristae. Okay. So the correct match for cristae will be infoldings in mitochondria. Okay. Now, another important organelle that is found specifically in plants and euglenoids are plastids. These are responsible for giving specific colors to the plant cells, and one of the most common examples of plastids is chloroplast, which gives green color to the plants. So this chloroplast is also a double membrane structure, and the space that is limited by the inner membrane is known as stroma, and this stroma consists of various flattened membranous sacs known as thylakoids, which are arranged just like piles of coins known as grana. Okay, so the correct match for the thylakoid will be flattened membranous sacs in stroma of plastids. That is number 4. Okay, now next one is centromere. So we know that a chromosome has a primary constriction which binds the two chromatids together. Okay, so this primary constriction is known as centromere. Okay, So the correct match for centromere will be number 1, that is primary constriction in chromosome. Okay. And lastly, we have an important organelle known as Golgi apparatus, which is responsible for packaging of materials, and it consists of many flat disc-shaped structures known as cisternae. Okay. This is an important site for the formation of glycolipids and glycoproteins. So the correct option for cisternae will be disc-shaped sacs in Golgi apparatus. So the correct option will be option B. Okay. So I hope you understood. Question number 22 is from the chapter Morphology of Flowering Plants, class 11. And the question is, diadelphous stamens are found in, and the options are China rose, citrus, P, China rose and citrus. And the correct option is option C, that is P. So what is a stamen? A stamen represents a male reproductive organ of a flower, and it consists of two different parts, namely anthers and filament. Now many stamens together constitutes an androecium. Now these stamens can be arranged in different forms. For example, these can either remain free or they can be united into bundles. Okay. Now depending on the number of bundles the stamens are united into, they are classified as monoadelphous, diadelphous and polyadelphous. Okay. So as the name suggests that monoadelphous are those stamens which are united into one bundle. Okay. Similarly, diadelphous are those stamens which are united into two bundles, and polyadelphous stamens are united into many bundles. Okay. Now let's see example of each of them. One of the very common example of monoadelphous stamens is China rose. The most common example for diadelphous stamen is P, and the example for polyadelphous stamen is citrus or bombex. Okay. So from the given option, you can see that P is the example of diadelphous stamens, China rose is the example of monoadelphous stamen, and citrus is the example of polyadelphous stamen. Okay, I hope you understand. Question number 23 is from the chapter Strategies for Enhancement in Food Production, class 12, and here we have to match list 1 with the correct option in list 2. So this question is related to the topic tissue culture. What is tissue culture? It is basically a technique through which we can grow a whole plant body using a part of the plant or cell of a plant which is taken and grown in a test tube under special condition, right? That means this cell or part of the plant has the potential to grow into a whole plant body. So this potential or capacity is known as totipotency. Okay. So the word totipotency is related to plant tissue culture. Okay. Now, one of the applications of tissue culture is somatic hybridization. Have you heard of the word somatic hybridization? In this technique, we take a plant cell, and as we know that a cell of a plant or a bacterium consists of a cell wall. So during this process, this cell wall is removed by digesting it. So what is left is a live cell without a cell wall, that is known as protoplast. Okay. Now if we take two different protoplasts from two different varieties of plants and fuse them to get a hybrid variety, then it is expected that it will exhibit a unique property. Okay. So one of the imaginary hybrid is pomato. What is pomato? It was imagined that if we fuse the protoplast of a tomato and the protoplast of a potato, it will lead to the formation of pomato with some desired characteristics. But this experiment failed because the resulting species was just the tomato. Okay. Now, another application of tissue culture is micropropagation. What do you understand by micropropagation? It is basically a technique through which a large number of plants can be grown using tissue culture. Okay, and one of the special features of this micropropagation is that all the plants are genetically identical to the original plant, right? And these plants are said to be the soma clones of the original plant. Okay, so the word soma clones is related to micropropagation and pomato is related to protoplast fusion. Okay, now what is left? Virus free plant. So the technique used to get a virus free plant is meristem culture. Why meristem culture? The reason is that even if a plant has been infected by a virus, its meristem remains unaffected. Okay, so we take the meristem in test tube and grow it in vitro to get a plant that is virus free plant. Okay, so the virus free plants is related to meristem culture. So the correct option out of the four given options will be option B. Got it? I hope you understood. Question number 24 is from the chapter Organisms and Populations, class 12, and the question is amensalism can be represented as. So here we have to remember that whenever there is an interaction between any two organism or species, then this interaction can be a positive interaction, it can be a negative interaction, or it can be a neutral interaction for one or both the species. Okay. So in case the interaction is positive for any of the organisms, then the organism is said to be benefited by this interaction, right? If the interaction is negative, then the organism is said to be harmed by this interaction. And if the interaction is neutral, that means the organism remains unaffected. Okay. So what happens in case of amensalism? In this case, one of the species is harmed whereas the other species remains unaffected. And how does it happen? See, one of the examples is, suppose this is the species 1 and it secretes certain substances. Okay, and its secretion is able to inhibit the growth of other species. Okay, then in that case, this species is harmed whereas the first species remains unaffected because it is neither benefited through its secretion nor harmed. Okay, so in this case option A will be the correct option. Okay, what does option B denotes? See, species A is benefited whereas species B is also benefited. That means both the species are benefited. So this is a case of mutualism where both the species help each other in some ways. Okay. Now in option C, species A and species B both are harmed. This is a case of competition. Okay. And option D shows that the species A is benefited whereas species B remains unaffected. That means it is just the opposite of option A, that is opposite of amensalism, that is commensalism. Okay, that means what happens in commensalism? In this case, one is benefited whereas the other is unaffected. Okay, so I hope you understood this question. Question number 25 is from the chapter Anatomy of Flowering Plants, class 11, and the question is, which of the following is an incorrect statement? Option A, mature sieve tube elements possess a conspicuous nucleus and usual cytoplasmic organelles. Option B, microbodies are present both in plants and animal cells. Option C, the perinuclear space forms a barrier between the materials present inside the nucleus and that of the cytoplasm. And option D, nuclear pores act as passages for proteins and RNA molecules in both directions between the nucleus and cytoplasm. So the incorrect statement out of the given four statements is option A. Okay. So let's see what are sieve tube elements. Okay. So we know that vascular bundles in plants are of two types. Xylem, which is responsible for the transportation of water and minerals, and phloem, that is responsible for the transportation of food particles. Okay. Now this phloem has four important parts. Okay. These are sieve tube elements, companion cells, then phloem parenchyma and phloem fibers. Okay. So now we know that sieve tube elements are part of phloem, right? Now these sieve tube elements remain in association with special type of cells known as companion cells. Okay, these companion cells consist of nuclei, but the sieve tube elements do not contain nuclei. Okay, so these sieve tube elements are arranged longitudinally with the companion cells. So can you identify? This is the companion cell, whereas this is the sieve tube element. Okay. The end wall of the sieve tube elements are perforated in a plate-like manner to form sieve plates. Now, initially, the sieve tube elements have all the organelles present in the cytoplasm. Okay, but once they start maturing, the cytoplasm moves to the periphery and these organelles are lost. Only few organelles are left. Okay, so the sieve tube elements have peripheral cytoplasm. Okay, and a large vacuole but no nuclei. Okay, so who controls the action of sieve tube elements? These actions are controlled by the nucleus of companion cell. Okay, therefore these sieve tube elements do not have their own conspicuous nucleus. Now, next option is microbodies are present both in plants and animal cells. This is a correct statement. Option C, the perinuclear space. So what is a perinuclear space? The space around the nucleus, right? So this space, as you can see here, acts as the wall of separation between the nucleus and the cytoplasm. So it acts as a barrier between the materials present in the nucleus and that in the cytoplasm. Okay. Now option D is nuclear pores act as passages for proteins and RNA molecules in both directions between nucleus and cytoplasm. This is also a correct statement. Okay. Question number 26 is from the chapter Sexual Reproduction in Flowering Plants, class 12. And the question is, a typical angiosperm embryo sac at maturity is, and the options are 8 nucleate and 7 celled, 7 nucleate and 8 celled, 7 nucleate and 7 celled, and 8 nucleate and 8 celled. So the correct option is option A, that is 8 nucleate and 7 celled. So let's see what is an embryo sac. We know that the ovule in a plant consists of one or two protective covering which are known as integuments. So the outer covering is known as outer integument, whereas the inner covering is known as inner integument. Also, the ovule is attached to the placenta by means of a stalk known as funicle. As you can see here, the integuments encircle the ovule on all sides except at the tip, and this is known as micropyle. So this end of the ovule is known as micropylar end. Opposite to this micropylar end is the chalaza, which represents the basal end of the ovule, right? Now, enclosed within these integuments is a special mass of cell known as nucellus, which is abundant in food reserve. Now, within these nucellus, you will find the female gametophyte, which is known as embryo sac. So now we know what is an embryo sac and where is it located, right? Now let's see how is embryo sac developed. So the development of embryo sac starts with the formation of megaspores from the megaspore mother cell. How does this happen? The ovules differentiate these megaspore mother cell in the micropylar region of the nucellus. Okay, then these megaspore mother cell undergo meiotic division. What happens in meiotic division? 4 nuclei is formed. So it leads to the formation of 4 nuclei out of which only one remains functional, whereas the other three are degenerated. Okay, so this is the functional nuclei that leads to the development of embryo sac. Okay, now the nucleus of this functional megaspore divides mitotically into two nuclei. Okay, and these two nuclei move to the opposite pole. Two more sequential mitotic divisions take place, and so one mitotic division here, so two nuclei, and one mitotic division here, again two nuclei. One more mitotic division will take place, so again one mitotic division here, so we'll get four nuclei here and four nuclei at the chalazal end. Okay, so it leads to the formation of four nucleate stage and eight nucleate stage. What happens after this 8 nucleate stage? See, 3 nuclei remain at the micropylar end and 3 remain at the chalazal end, whereas 2 move to the central cell. So these 2 remain in one cell. Okay, the nuclei at micropylar end are divided into 3 different cells. Okay, 2 are synergids, whereas 1 is egg cell. Okay, similarly, 3 nuclei are situated at the chalazal end. So how many nuclei are there? 3 nuclei here, 3 nuclei here, and 2 nuclei at the center. So total number of nuclei are 8, right? And how many cells are there? So 3 cells here, 3 cells here, and 1 cell in the center. So total 7 cells. So what will be the answer? 8 nucleate and 7 celled. Okay. Question number 27 is from the chapter Plant Kingdom, class 11, and the question is, which of the following algae contains mannitol as reserve food material? The options are Ectocarpus, Gracilaria, Volvox and Eulothrix. So the correct option is option A, that is Ectocarpus. Okay. So what are algae? Algae are photosynthetic, mainly aquatic and thalloid organism. What do you mean by thalloid? That means they lack any true root, stem, leaves or any reproductive structures. Okay. Now these algae are classified into three main classes. Okay. These are chlorophyceae, which are also known as green algae. Then phaeophyceae, which are also known as brown algae. And lastly, rhodophyceae, which are also known as red algae. Okay. Now all these algae have different reserve food materials. So the reserve food material in case of chlorophyceae or green algae is starch. The reserve food material in case of phaeophyceae is mannitol or laminarin. Okay. And the reserve food material in case of rhodophyceae are fluoridine starch. So according to this question, the mannitol is the reserve food material in one of the phaeophyceae, right? And Ectocarpus is the example of phaeophyceae. What is Gracilaria? It is an example of rhodophyceae or red algae, whereas Volvox and Eulothrix are example of chlorophyceae or green algae. Question number 28 is from the chapter Plant Growth and Development, class 11, and the question is, the plant hormone used to destroy weeds in a field, and the options are indole acetic acid, naphthalene acetic acid, 2,4-D or 2,4-dichlorophenoxyacetic acid, and indole butyric acid. So the correct option is option C, that is 2,4-dichlorophenoxyacetic acid. So all the given four options are types of auxins. What are auxins? These are plant hormones which are produced at the growing apices of stems and roots. Okay, and from these locations they migrate to their regions of actions. Okay, now these auxins are of two types, natural and synthetic auxins. Okay, out of these, only some of the synthetic auxins can be used as weedicides to destroy weeds. Now, out of the given four options, indole acetic acid and indole butyric acid are types of natural auxins since they have been isolated from plants. So they cannot act as weedicides. Okay. Now, out of naphthalene acetic acid and 2,4-dichlorophenolic acid, only 2,4-D is used as weedicides. Okay. And it is widely used to kill dicot weeds without affecting any monocot plant. Okay, therefore it helps in preparing weed free lawn without harming any useful plant. Question number 29 is from the chapter Ecosystem, class 12, and the question is the amount of nutrients such as carbon, nitrogen, phosphorus and calcium present in the soil at any given time is referred as, and the options are climax, climax community, standing state and standing crop. So the correct option is option C, that is standing state. Okay, so as you can see here, the question has been asked about the amount of nutrients. That means the amount of inorganic matter present in the soil at any given time. And this is known as standing state. Okay, so what is the difference between standing state and standing crop? The only difference is that standing state talks about the inorganic matter, whereas standing crop talks about the organic matter or living materials present in different trophic levels at any given time. Okay. What is climax? Climax means the most important point in any event. Okay. Now, what is a climax community? See, we know that our environment keeps on changing, so the species composition in an ecosystem also keeps changing with respect to this environment in a sequential manner. Okay, and finally it leads to a community that is in equilibrium with the environment. Okay, and this community is known as climax community. Okay. Question number 30 is from the chapter Plant Growth and Development, class 11, and the question is mutations in plant cells can be induced by, and the options are kinetin, infrared rays, gamma rays and zeatin. So the correct option is option C, that is gamma rays. So what do you mean by mutation? Mutation is any change in the DNA sequence or gene of an organism that leads to the formation of variant. That means a different variety of the same organism or species. Okay. Now the agents that lead to mutation are known as mutagens. Okay. Now these mutagens can be of three types: physical mutagens, chemical mutagens and biological mutagens. Okay, physical mutagens involve different kind of radiations, for example x-rays, gamma rays, uv rays, etc. Okay, so gamma radiation or gamma rays is the correct option. Okay, now what do you mean by kinetin? Kinetin is a type of synthetic cytokinin hormone. Okay, and it was obtained from degraded product of autoclave herring sperm, right? Whereas zeatin is a natural cytokinin and it was obtained from the unripe maize grain. Okay. Now what is infrared rays? These are the rays which cause heating effect. Question number 31 is from the chapter Ecosystem, class 12, and the question is which of the following statements is not correct, and the options are pyramid of biomass in C is generally inverted, pyramid of biomass in C is generally upright, pyramid of energy is always upright, and pyramid of numbers in a grassland ecosystem is upright. So the incorrect statement out of the given four is statement B. Why? Because the pyramid of biomass in C is not upright, it is inverted in shape. So you must be familiar with the shape of a pyramid. How does it look? It has a broader base and a narrow apex, right? So if we try to find out the relationship between organisms and their numbers, biomass or energy at different trophic levels, we get a similar kind of shape. Therefore, this relationship is known as ecological pyramid. Here, the first trophic level represents the plants or autotrophs which can produce their own food. Therefore, these are known as primary producers, and these do not depend on any other organisms for food. Okay, but from the next trophic level, dependency starts. That means the next trophic level, which includes primary consumers, depend on the primary producers for food. Similarly, the next trophic level include secondary consumer, and they depend on the primary consumer for food. And the last one is tertiary consumer, which depend on the secondary consumer for food. Okay. So here you can observe that the first trophic level has a large width, whereas the next trophic level has a smaller width. Okay, so why does the width keeps decreasing? See, take an example. Suppose plants are the primary producers. Okay, so can one goat eat more than one plants? Yes, one goat can eat 4 to 5 plants. Okay, that means the number of goat is smaller than the number of plants. So the trophic level representing the goat will be smaller in width, okay, than the plants. Similarly, more than one goats, you can say 4 to 5 goats can be eaten by just one lion. So the next trophic level, which represents a carnivores or lion, will be smaller in width than the number of goats. So this is the pyramid of number. Okay. So the number keeps on decreasing, therefore the width also keeps on decreasing as we move upward. Okay. This is the case with the pyramid of biomass also. The biomass of goat will be less than the biomass of 4 to 5 plants. Similarly, biomass of one lion will be less than the biomass of 4 to 5 goats. Okay. But in case of aquatic environment, this is not always the case, especially when the primary producers are phytoplanktons and the primary consumers are zooplanktons. Okay. Why? Because here the number of zooplankton is more than the number of phytoplanktons. Okay, so suppose this is one phytoplankton in water, then more than one, in fact hundreds or thousands of zooplanktons will feed on this plant. Okay, therefore the number of zooplankton will be larger than the number of plants, so it will look inverted in shape. Okay, also the biomass of zooplankton will be more than the biomass of phytoplankton. Okay, therefore the pyramid of biomass in sea is not upright, it is inverted in shape. Okay, pyramid of energy is always upright. This is correct statement. Why? Because energy always keeps on decreasing. Only the energy left in the plants will get transferred to the next level, and the only part of that energy will get transferred to the next. Okay, the energy can never be transferred which is more than the previous energy. Okay. Question number 32 is from the chapter Ecosystem, class 12, and the question is, in the equation GPP minus R equals to NPP, R represents, and the options are radiant energy, retardation factor, environmental factor, and respiration losses. So the correct option is option D, that is respiration losses. Now in order to understand this, we first must understand what is an ecosystem. Okay, so we know that our environment consists of living organisms known as biotic components, as well as non-living organisms known as abiotic components. Right? Now these components keep interacting among themselves as well as with their environment. So this interaction of biotic and abiotic components among themselves and with their environment is known as ecosystem. Now, the basic requirement for any ecosystem to function and sustain is the constant input of solar energy. Why? Because plants use solar energy to produce food through the process known as photosynthesis. Now, the amount of this food produced in terms of biomass per unit area is known as primary production. Okay, and the rate of the biomass production is known as productivity. Now this productivity is of two types: gross primary productivity and net primary productivity. What is the difference between these two? See, gross primary productivity is the rate of production of organic matter during photosynthesis. Okay, but a considerable amount of this GPP is lost during the respiration. So what is left after this respiratory loss is known as net primary productivity, which is left for consumption by the herbivores. Okay, so here R represents the respiratory loss or respiration loss. Okay, therefore option D is the correct answer. Question number 33 is from the chapter Plant Kingdom, class 11, and the question is, which of the following algae produce carrageenan? And the options are green algae, brown algae, red algae and blue green algae. So the correct option is option C, that is red algae. Now what are algae? Algae are simple thalloid and photosynthetic or autotrophic organisms which are found largely in water. Okay, now these algae are broadly classified into three types: chlorophyceae or green algae, phaeophyceae or brown algae, and rhodophyceae or red algae. Okay, now the algae are very important because they are photosynthetic in nature, therefore they help in fixing carbon dioxide in the environment and they increase the amount of dissolved oxygen in their immediate environment. Other than these, some economically important substances are obtained from the cell wall of algae. Okay, for example, carrageenan is one such substance which is obtained from the cell wall of red algae. Other than carrageenan, the cell wall of red algae also contains agar and fumarate. Okay. The cell wall of brown algae contains algin, which is also economically important substance. Okay. What does green algae have in their cell wall? They are composed of cellulose and pectin. Now, blue-green algae have mucopeptides in their cell wall. Okay. Question number 34 is from the chapter Photosynthesis in Higher Plants, class 11, and the question is, the first stable product of carbon dioxide fixation in sorghum is, and the options are pyruvic acid, oxaloacetic acid, succinic acid and phosphoglyceric acid. So the correct option is option B, that is oxaloacetic acid. So we know that plants use the process of photosynthesis to produce their food, right? Now the photosynthesis consists of two types of reactions: light reactions, which are directly light driven, and dark reactions, which are not directly light driven but depend on the products of light reactions. Okay. Now two different groups of plants exist, C3 and C4 plants, which use different pathways for dark reaction of the photosynthesis. The main difference between C3 and C4 plants is that the first stable product of carbon dioxide fixation in C3 plants is 3-phosphoglyceric acid, whereas that in C4 plants is oxaloacetic acid, right? Most of the plants belong to the C3 group. Some of the examples are rice and wheat, whereas some of the plants such as maize and sorghum belong to the C4 plants. Therefore, the first stable product in case of sorghum will be oxaloacetic acid. Okay. Now, what is pyruvic acid? It is the end product of glycolysis, whereas succinic acid is the intermediate product of Krebs cycle. So these two are not related to the photosynthesis. Okay, phosphoglyceric acid is related to photosynthesis but in C3 plants and oxaloacetic acid in C4 plants. So I hope you understood. Question number 35 is from the chapter Anatomy of Flowering Plants, class 11, and here we have to match list 1 with the correct option in list 2. So the first one is lenticels. What are lenticels? These are lens-like openings in the stems of plants which allow the exchange of gases. So the correct match for number A will be exchange of gases, that is number 3. Okay. Now what is cork cambium? In order to understand this, we should know that growth in plants are of two types, primary growth and secondary growth. What is primary growth? It involves the increase in length of roots and stems with the help of apical meristem. On the other hand, secondary growth involves increase in girth of bark of trees with the help of lateral meristem. Now, this lateral meristem is of two types, vascular cambium, which develops first, and cork cambium, which develops at later stage. Okay, this cork cambium is also known as phelogen. So the correct match for cork cambium will be number 1, that is phelogen. Okay, now this phelogen cuts the cells on both sides. For example, this is the cell and this is the phelogen. Shaded portion is phelogen. So it is cutting the cell in two parts, the inner part and the outer part. Okay, so the outer part differentiate into cork, and this cork has a deposition of suberin in their cell wall. Therefore, the correct match for cork will be suberin deposition, that means number 2. Whereas the inner part develops into secondary cortex, and this is also known as pheloderm. Okay. Therefore, the correct match for secondary cortex will be pheloderm. Okay. So the option B is correct option. Okay, I hope you understood this. Question number 36 is from the chapter Respiration in Plants, class 11, and the question is, which of the following statements is incorrect? So the options are, during aerobic respiration, role of oxygen is limited to the terminal stage. In electron transport chain, one molecule of NADH plus H plus gives rise to 2 ATP molecules and one FADH2 gives rise to 3 ATP molecules. ATP is synthesized through complex 5, and the last option is oxidation reduction reactions produce proton gradient in respiration. Okay, so the incorrect option is option B. Okay, let's see what is an electron transport chain. It is a chain or system which takes place in the inner mitochondrial membrane and involves the oxidation of NADH plus H plus and FADH2 molecules to synthesize ATP molecules. Okay. What happens during electron transport chain? Electrons are passed from one carrier to next through 5 complexes. Okay, these are complex 1, 2, 3, 4 and complex 5. Okay, what is complex 1? It is NADH dehydrogenase. Okay, complex 2 is FADH2. Complex 3 is cytochrome BC1, whereas complex 4 is cytochrome C oxidase, and lastly complex 5 is ATP synthase, and this is the most important complex. Okay, now while passing through the carriers, electrons are coupled to this complex 5 or ATP synthase to produce ATP molecules from ADP and inorganic phosphate. Okay. Now the number of ATP produced depends on the nature of electron donor. For example, one molecule of NADH gives rise to 3 molecules of ATP, whereas one molecule of FADH2 gives rise to 2 molecules of ATP. Therefore, option B is incorrect. Okay, because here it is written that NADH gives rise to 2 ATP. It's false because it gives rise to 3 ATP molecules. Whereas one FADH2 gives rise to 2 ATP molecules, not 3 ATP molecules. Okay. Now, at last what happens? Oxygen acts as the final electron acceptor and then it removes this electron from the system. So the role of oxygen is limited to the last stage or the terminal stage. Okay. Now, Option A, here we see that role of oxygen is limited to terminal stage. This is true. We saw here. Now, option B was false. Option C, ATP is synthesized through complex 5. This is also true. We saw here. And last is oxidation reduction reactions produce proton gradient in respiration. This is also true because according to chemiosmotic hypothesis, the ATP is synthesized after the formation of a proton gradient which is established during the oxidation reduction reaction. So I hope you understood this. Question number 37 is from the chapter Morphology of Flowering Plants, class 11. And here we have to match the floral formula of flowers with the family to which the flowers belong. Okay, so we know that various morphological features are used to describe a flowering plant, and it is required that the description should be simple, scientific, and in a proper sequence. So one of such descriptions is used to represent the structure of particular type of flowers using the formula known as floral formula. Okay. Now various signs or symbols are used in floral formula. For example, BR stands for bracteate. K stands for calyx. C stands for corolla or petals. Okay. Then P stands for perianth. A stands for andrecium. G stands for gynoecium. Then if you have a straight line above G, it represents superior ovary, whereas a line below G represents an inferior ovary. Okay. Then you have some male signs. You must be aware that this is represented as this. Okay. Similarly, female sign is this, and the combination of both represents the bisexual organisms. Okay. Similarly, symmetry can also be shown. For example, this sign indicates actinomorphic or radial symmetry. Okay. Similarly, a sign like this represents bilateral symmetry or zygomorphic. Okay. Now, look at the options given here. The first option is where there is a zygomorphic symmetry. Okay. And out of the given options, the correct match for this is Fabaceae. Okay. Now, the next one is here calyx, corolla and andrecium all are 5 in numbers. Also, corolla and andrecium are fused together. So, this is the characteristics of Solanaceae family. Okay. In the third one, you can see perianth and andrecium are fused together, and this is the characteristic of Liliaceae. Similarly, the last one is the floral formula for Brassicaceae. Okay, so the correct option would be option A, which shows the correct match for the floral formula and the families. So I hope you understood about the floral formula. Question number 38 is from the chapter Cell Cycle and Cell Division, class 11, and here we have to match list 1 with the correct option in list 2. To understand this question, we should know about different phases of cell cycle. As we know that a typical eukaryotic cell divides once in every 24 hours, but this duration may vary depending on the cell type or type of organisms. Okay. But a normal cell cycle consists of two main phases. The first one is M phase, during which the actual cell division or mitosis takes place. Okay. So let's shade this area. And the rest of the duration includes 95% of the total duration, and it is the interphase. What is interphase? It is the phase between two successive M phases, right? Now this interphase is further divided into three phases. First one is G1 or gap 1 phase, second one is S phase or synthesis phase, and third one is G2 or gap 2 phase. Okay, let's see what happens during G1 phase. This is the phase during which the mitosis is just ended, okay, and the preparation for the DNA replication starts. So the correct match for G1 is interval between mitosis and initiation of DNA replication. What is S phase? This is the phase during which actual DNA replication is done. Okay, so the correct match for S phase is DNA replication. Now G2 phase, during this phase protein is synthesized. So what will be the correct match for G2 phase? Number 1, that is proteins are synthesized. Now the only one thing is left, quiescent stage. What is quiescent stage? See, sometimes cells do not divide. For example, our heart cells do not divide, and there are some other cells which divide only during an important event, for example during injury. So these cells undergo this cell division or meiosis. After this, they immediately enter into a state of inactivity. Okay, this one. This is known as G0 phase. During this phase, they remain metabolically active so that they can start dividing again as soon as the situation demands, but they do not divide further. Okay, unless and until the situation demands. Okay, so the correct match for option C will be inactive phase. So the correct option out of A, B, C, D will be option C. I hope you understood this. Question number 39 is from the chapter Biotechnology Principles and Processes, class 12. And the question is, plasmid PBR322 has PST1 restriction enzyme site within ampicillin resistance gene that confers ampicillin resistance. If this enzyme is used for inserting a gene for beta-galactosidase production and the recombinant plasmid is inserted in an E. coli strain, then the options are, it will not be able to confer ampicillin resistance to the host cell. The transformed cells will have the ability to resist ampicillin as well as produce beta-galactosidase. It will lead to lysis of the host cell and it will be able to produce a novel protein with dual ability. So the correct option is option A, that is it will not be able to confer ampicillin resistance to the host cell. Now let's try to understand the basic concept behind this question. We know that in recombinant DNA technology, we isolate a DNA fragment with some desired characteristics and ligate it to the vector DNA or plasmid DNA in order to transfer it to the host cell, right? But in order to transfer or ligate this DNA to the vector DNA, it is required that the vector should have preferably one recognition site. What is recognition site? The site at which the restriction enzyme cuts the DNA, and then at this position this DNA will be ligated. Okay. Now generally what happens, the ligation is carried out at restriction site present in one of the two antibiotic resistance gene. Okay, that means one of the antibiotic resistance genes are used. These are ampicillin resistance gene and tetracycline resistance gene. The recognition sites present in ampicillin resistance gene are PST1 and PVU1. Okay. Whereas tetracycline resistance gene has only one recognition site, that is BAMH1, right? But here we have to consider PST1 site of ampicillin resistance gene because this is mentioned in the question. Okay. Also, according to this question, we have to ligate an alien DNA, that is the DNA or gene for beta-galactosidase production, right, into the vector, and in this case the vector is PBR322, and then this has to be inserted into the host. What is the host here? E. coli. Okay. Now what will happen after inserting beta-galactosidase in the ampicillin resistance gene? It will get deactivated. That means the ampicillin resistance gene will get deactivated, and this is known as insertional inactivation or insertional deactivation because it has been caused due to the insertion of an alien DNA or gene. Okay. due to which this will not be able to confer the ampicillin resistance to the host cell. Okay, therefore option A is correct. Option B is the transformed cells will have the ability to resist ampicillin. So this is wrong because it will not have the ability to resist the ampicillin. Okay, but it will be able to produce beta galactosidase. Option C, it will lead to lysis of the host cell. This is not true. No lysis will take place. Option D is it will be able to produce a novel protein with dual ability. No, what is being produced? Beta galactosidase, right? But this is not a novel protein and it is not a dual, it does not have a dual ability as well. So option A is the only correct option. I hope you understood this. Question number 40 is from the chapter Molecular Basis of Inheritance, class 12, and the question is identify the correct statement. Option A, in capping, methyl guanosine triphosphate is added to the 3' end of the hnRNA. Option B, RNA polymerase binds with Rho factor to terminate the process of transcription in bacteria. Option C, the coding strand in the transcription unit is copied into an mRNA. And option D, split gene arrangement is a characteristic of prokaryotes. So the correct option is option B and rest are false. Okay. So we know that a DNA has two strands. One is template strand and the other is coding strand. During the process of transcription, this template strand is copied into an mRNA. Therefore, option C is false because the coding strand is not copied, the template strand is copied. Okay. Now, the enzyme used is DNA dependent RNA polymerase. Why this name? Because RNA is to be polymerized using the strand of DNA. Therefore, it is DNA dependent RNA polymerase. Now, the process in case of prokaryotes and eukaryotes are different. In case of prokaryotes or bacteria, the process is relatively simpler. Okay, there is only single RNA polymerase that performs the process of transcription. So, to initiate the transcription, the RNA polymerase enzyme binds to a factor known as sigma factor. Whereas to terminate the process after the transcription is complete, it binds to a factor known as Rho factor. Therefore, option B is correct because it binds to the Rho factor to terminate the process of transcription. Okay. Now, this is a simple process in prokaryotes. What happens in eukaryotes? Here there are some complexities, and one of the complexities is that the primary transcripts here consists of two types of sequences. Okay, one is exon or coding sequence and the other is introns or intervening sequences. What do introns do? These interrupt the exons. Okay, so this is known as split gene arrangement because the exons is split by the introns. Okay, split gene arrangement. Therefore, option D is false because it happens in case of eukaryotes, not prokaryotes. Okay. Now, so it is required that the introns should be removed and the exons should be joined together. And this is done by a process known as splicing, which consists of two steps. First is capping and the other is tailing. What happens in capping? Here methyl guanosine triphosphate is added at the 5' end of the hnRNA. Okay. So this is hnRNA. This is suppose 5' end. So methyl guanosine is added here. Therefore, option A is false because it is not added at 3' end. It is added at the 5' end. And during tailing, adenylate residues are added at the 3' end of the hnRNA. Okay. So I hope you understood this. Question number 41 is from the chapter Biotechnology and its Applications, class 12, and the question is, nowadays it is possible to detect the mutated gene causing cancer by allowing a radioactive probe to hybridize its complementary DNA in a clone of cells followed by its detection using autoradiography because option A, mutated gene partially appears on a photographic film. Option B, mutated gene completely and clearly appears on the photographic film. Option C, mutated gene does not appear on a photographic film as the probe has no complementarity with it. And option D, mutated gene does not appear on the photographic film as the probe has complementarity with it. So the correct option is option C, that mutated gene will not appear on the photographic film and the probe has no complementarity with it. Okay. So let's see what is a probe or a radioactive probe. It is a single stranded DNA or RNA that is tagged with a radioactive molecule. Okay, and this probe is allowed to hybridize to its complementary DNA in a clone of cells, followed by its detection using the technique of autoradiography. Okay, so what will happen? The clone having the mutated gene will not appear on the photographic film because the probe will not have the complementarity with the mutated gene. Okay, therefore option C is correct while the rest are false. Okay, I hope you understood this. Question number 42 is from the chapter Organisms and Populations, class 12, and the question is, in the exponential growth equation, nt equals to n naught e to the power rt, e represents, and the options are the base of number logarithms, the base of exponential logarithms, the base of natural logarithms, and the base of geometric logarithms. So the correct option is option C, that is the base of natural logarithms. Okay, so we know that for the growth of any population, the availability of resources is important, right? Now suppose the amount of resource in any habitat is unlimited. Then what will happen in this case? Each species will have the full potential to grow in number. That means they will grow very fast, or we can say they will grow exponentially. So this type of growth is known as exponential growth, right? Now suppose n is the size of a population, then this n will vary with time, right? And in case of exponential growth, what will happen? This n will keep on increasing, right? So if we have to plot a graph showing the relation of n with time, then it will give us a J-shaped curve which shows that the population is increasing continuously, right? And the integral form of this exponential growth can be expressed as nt equals to n naught e to the power rt, where nt represents the number density or population density after time t. Okay, n naught represents the population density at time 0, that means just at the starting point. E is the base of natural logarithms. R shows the intrinsic rate of natural increase. Okay. And what is T? It is time. Okay. So I hope you understood this. Question number 43 is from the chapter Anatomy of Flowering Plants, class 11, and the question is, select the correct pair. Option A, large colorless empty cells in the epidermis of grass leaves are subsidiary cells. So this is wrong because these are not subsidiary cells. These are known as bulliform cells. Okay. So option A is incorrect. Option B says in dicot leaves, vascular bundles are surrounded by large thick-walled cells, and it is mentioned as conjunctive tissue, which is wrong since these cells are known as bundle sheath cells. Okay. Therefore, option B is also incorrect. What does option C say? It says cells of medullary rays that form part of cambial ring are interfascicular cambium, and this is a correct statement. Let's see the concept. We know that growth in case of plants is of two types. First one is primary growth, whereas the other type is secondary growth, right? What is primary growth? We know that the increase in length of roots and shoots with the help of apical meristem is known as primary growth. Okay, whereas the increase in girth of the plant with the help of lateral meristem is known as secondary growth. Also, the lateral meristem is classified into two types. First one is vascular cambium, and this appears first, okay, whereas the other type of lateral meristem known as cork cambium appears later. So let's see about the vascular cambium. Suppose this is the transverse view of a dicot stem. So here you will see a large number of fascicles present here, okay. Now these fascicles have their inner part as the primary xylem, whereas the outer part is primary phloem, okay. So what happens in the young stem, vascular cambium is present in patches as single layer between the xylem and phloem. You can see here this layer is of vascular cambium, okay. But later on, these cells of medullary rays join together and form a ring, okay. So this is known as interfascicular cambium, whereas the earlier cambium was known as intrafascicular cambium. Why? Because it was present inside the fascicles, whereas interfascicular cambium are now present in between two fascicles, okay. Therefore these are known as interfascicular cambium. So option C is a correct option, okay. Now, what does option D say? Loose parenchyma cells rupturing the epidermis and forming a lens-shaped opening in the bark is spongy parenchyma. So this is also false because these are not spongy parenchyma. These are known as complementary cells. Okay. Question number 44 is from the chapter Sexual Reproduction in Flowering Plants, class 12. And the question is, in some members of which of the following pairs of families, pollen grains retain their viability for months after release? That means they remain viable for a long time. So the options are Poaceae and Rosaceae, Poaceae and Leguminosae, Poaceae and Solanaceae, and Rosaceae and Leguminosae. So the correct option is option D, which contains the pair Rosaceae and Leguminosae. Okay. Now we know what happens during fertilization. During this process, the pollen grains from the flowers are transferred to the stigma of flowers. Now these pollen grains have to land on the stigma before they lose their viability. What is viability? It is their ability to carry out the fertilization. Okay. Now the period for which the pollen grains remain viable depends on prevailing temperature and humidity to some extent, but most importantly, it varies from one family to another. For example, some families like Rosaceae, Leguminosae and Solanaceae, the pollen grains remain viable for about a month as asked in the question. But the families like Poaceae, they do not remain viable for a long time. In fact, they remain viable only for few minutes. Okay, so they lose their viability within 30 minutes. So all the options containing Poaceae cannot be the correct option. Okay, so only correct option will be option D. I hope you understood this. Question number 45 is from the chapter Molecular Basis of Inheritance, class 12. And the question is, what is the role of RNA polymerase 3 in the process of transcription in eukaryotes? And the options are transcribes rRNAs 28S, 18S and 5.8S. Then transcribes tRNA, 5S rRNA and snRNA. Transcribes precursor of mRNA or transcribes only snRNA. So the correct option is option B, that is it transcribes tRNA, 5S rRNA and snRNA. Okay. Now we know that the copying of genetic information from one strand of a DNA to an mRNA is known as transcription, right? Now this transcription takes place in different ways in case of prokaryotes and eukaryotes. What happens in prokaryotes? Here the transcription takes place in the same compartment, okay, and the process is quite simple, and only one RNA polymerase enzyme is involved in the process, okay. Whereas in case of eukaryotes, first of all, the transcription and translation takes place in two different compartments. Then here the process of transcription is quite complex with the involvement of at least 3 RNA polymerase enzymes present in the nucleus in addition to the RNA polymerase present in the organelles. Okay. So this makes the process quite complex here. So out of the 3 RNA polymerase enzymes present in the nucleus in case of eukaryotes, the RNA polymerase 1 is responsible for the transcription of rRNAs. Okay, which rRNAs? 28S, 18S and 5.8S. Therefore, the first option is related to RNA polymerase 1 enzyme. Okay. RNA polymerase 2 enzyme is involved in the transcription of precursor of mRNA. What is the precursor of mRNA? It is hnRNA. Okay. Therefore, option C is related to RNA polymerase 2, whereas RNA polymerase 3 is responsible for the transcription of tRNA, 5S rRNA and snRNA. Therefore, option B is the correct option. Okay. So, I hope you understood this. Thank you.